Hi, everybody. Welcome back. This is chapter 16. We're going to be getting into the acquired immune stuff. So like specific immunity responses to specific antigens by specialized cells. So let's get into that. These are your learning outcomes for this chapter, if you're interested again. Adaptive immunity, I'll call it adaptive immunity. I'll call it acquired immunity. Specialized immunity, all of these things mean the same thing. This is the line of immunity that's going to be attacking a pathogen or something foreign, usually a pathogen or its toxin, associated toxin, and attacking that specific thing and not having the innate general response. And that's the idea there. Lymphocytes are the cells that are going to be doing all this work. Immunocompetence is the term that's referring to your body being able to react to the many antigens that you can potentially see within a lifetime. So what is an antigen? It's a marker, a molecule that we can be seen as foreign and essentially reacted to by our immune system. So it is possible for an antigen to not provoke a response after being sensed by the immune system. And then immunogens is basically a term applying to the antigens that provoke an immune response. So the ones that we're actually caring about. If it has any potential to be reacted to by any aspect of that immune system, we would call it an antigen. If it can provoke a true immune response and stimulate the T and the B cells, then that is what we call an immunogen, but we'll be using that word pretty interchangeably in this chapter. Protein and polysaccharide molecules tend to be the biggest target here, and most of the time our own antigens don't provoke a response, but we've talked and mentioned previously about autoimmune disorders, and that is going to tie in with that, and we'll get into that in another chapter as well. HAMPs versus immunogens. HAMPs are those pathogen-associated molecular patterns, the sort of like peptidoglycan or LPS or whatever it is. Those things are general for like bacteria or gram-negative or whatever stupid thing you can think of there. That's super generalized, not for this place. So we're talking about immunogens. We're talking about really breaking it down to specific aspects of our bad guys and focusing on those specific things, because we have to be specific in order to really get our immune system interacting appropriately and not just generally all over the place. We don't want to be spitting out antibody every time we see a gram-negative bacteria. That's just nonsense. So immunogens are more specific and therefore more controlled as well. Basically, we're going to be talking about its high specificity, like an antibody only binds with its one antigen. It's not going to be binding with anything else. And it has actually a specific epitope, which is usually a part, like an actual molecular almost sentence of the whole book. So we call the whole book the antigen. Antibodies can interact with specific sentences within it that we call epitopes, which are very specific parts of those antigens. So that's how specific this is going to be getting. Then we have memory, which is essentially the cells that have been activated to respond to these sorts of immunogens or antigens. Once we have been triggered to respond to them, we will also set aside some that will wait for the next time so that we don't have to go through this process all over again. The stages of immunologic development here, basically lymphocytes, the white cells, the T, what are going to be the T and the B cells, right? And we're going to have presentation of antigens that's going to help activate and stimulate activation of these cells. We'll be challenging those activated cells with, those antigens to make sure that we're responding appropriately. And then we're going to start producing antibodies and or cell-mediated immunity, depending on if you're B or T cells, respectively. So this is the stage of lymphocyte development and function. We start off with our basic lymphocyte. So this is our lymphocyte, and here we will hopefully be going through clonal deletion. That's removing the cells that would respond to self molecules, okay? When we differentiate into B cells, we're going to continue our maturation in the bone marrow, whereas, and then we'll eventually move to lymph node and spleen in the periphery. Whereas in the T cell, we move first to the thymus for maturation and then do the same thing after we've matured there. This is a little schematic of the activation of these lymphocytes. So if we start over here, we have our antigen here. We have our markers on the outside of our antigen presenting cell. It is a phagocytic cell. We've taken up this antigen, the bacteria maybe, and it will eat it and present it on its little surface out here. See how the little, that is annoying. See how the little MHC markers are holding on to the little antigens there, holding around on the surface. That is so that they can interact with these T helper cells. So these are T helper cells interacting with that dendritic cell. So now they're being activated so they can go on to, differentiate and get multiply, proliferate, that's what I'm looking for, and then differentiate. And so now they're getting activated to do their job. So they're being activated, and now they are going to make memory cells and activation of cytotoxic T cells. This is its memory cell. Everybody gets a memory cell when they get activated, FYI. I'm just going to follow their line down and go back to the B cells. We've got our activated T helper cell that's activating our cytotoxic cell, T cell. Yeah, it is what it sounds like. This is our T cells that are going to go out and kill infected cells, our cells, your cells. When you are sick with a virus, these are the T cells that are going to go to your cells that are, infected with the flu virus and kill them because they shouldn't be there. So it's the best way to get rid of it. So that's what these guys do. They're cytotoxic T cells. We also have regulatory T cells that are going to be involved in this whole process. Guess what they do? And then we have the T helper cells. Okay, well, cool. So they helped activate the cytotoxic T cell. Who cares? Well, okay, if you think that's not important, then you really just get ready. So here we have our B cells. They can often interact with antigen as well, but no matter what it is, at some point they're going to have to interact with another cell that also has the same antigen with it in order to get activated. So in this case, we have the helper T cell from way down at the bottom on the other side. We have that helper T cell that we had activated. It can come and help stimulate true activation of this B cell. When B cells are activated, they will proliferate and then differentiate as well. Proliferate, a whole bunch of B cell, general B cell, and then differentiate, some of them becoming plasma cells, some of them becoming memory cells. That's what I mean by proliferate and then differentiate. All right, so our activated B cell becomes plasma cells and memory cells. Memory cells, we already talked about what they do. They'll come out whenever we see this thing again. The plasma cells are going to be secreting antibodies. So plasma cells are actually pumping and pumping and pumping out antibody. It is all they're going to be doing. It is all they care about. It is all they will ever do. Making antibodies, secreting antibodies. They cannot do that until the B cell that they were before gets activated by usually a T helper cell that's already been activated with that same antigen. Okay, so all that has to happen together. So we make our little antibodies, and the antibodies can actually enter the bloodstream and move to other tissues, nearby tissues, and other stuff like that. So that's the gist of those cells, the lymphocytes. Okay, so let's really get into it. Markers on the surfaces of our cells will allow us to see our cells as self. And if they're lacking or have different markers, then we see it as non-self. And there has to be some sort of system in place, essentially, to help us differentiate between self, foreign, bad, attack it, and this isn't self, but we shouldn't attack it. So there has to be some sort of process in place in order for us to manage all of that. And there is. So we'll talk a little bit about that. We've already mentioned clonal deletion, and when these T or B cells that are in development early on in the bone marrow, whenever they recognize a self marker, they'll get deleted. They'll get killed. A major histocompatibility complex is actually really important as far as if you're having organ transplants. So all the cells have some kind of MHC marker on the outside. They're also known, and when I worked in the transplant lab, we called it HLA, which is human leukocyte antigen. It is a marker that in the transplant lab we used to determine similarities between the donors and the recipients. So it was these markers that we were looking at predominantly for similarities to make sure that somebody was not likely to reject an organ, which, you know, as soon as you're wrong, you know, whenever everything looks like it's 100% match, because there are other factors involved than just that. But basically, everybody has these on their surface of the cell. You can look at them as a marker of self because everybody's is different from one another. Or you can just look at them as these sorts of, what do you call them? They're not really receptors, but they're proteins that are going to display antigens that they've encountered on the surface of whatever appropriate cell, whether that be proper antigen presenting cells or just a cell like in your lungs that got infected with a virus. So they could actually present it on theirs as well. So the class one MHC, MHC1, these are on all nucleated cells. You need to know this. Display unique characters of self, so identifiers of self versus you're not me and I am not you. And then yes, and then each human inherits a particular combination of class 1 MHC genes. So yeah, everybody's different, and we have to try to match those genes up as best as we can, as predictably as we can, whenever we are trying to find an organ for somebody that needs an organ. Class 2 MHC, we have these on our antigen presenting cells, our proper professional antigen presenting cells, macrophages, monocytes, dendritic cells, B cells. That's all we're talking about here. They will go out and eat up a bacteria that came from outside of that cell. They eat it up and then break it down and present it out on the MHC2 genes. And that says this came from outside of the cell whenever it goes and interacts with T cells. If it goes and interacts with T cells, but it had come from inside itself, like whenever you have the flu and the cells in your lungs it could be any cells, right? Then it comes from inside of the cell and they're presenting it on MHC class 1 molecules, that would trigger a different reaction, because dealing with something outside of the cells versus something that lives inside of the cells is very different. But we do want to keep in mind that a lot of times whenever we are reacting to a bad guy, a pathogen, that we want to have both reactions in some degree. Now, one we might want stronger than the other though. Class 3 MHC genes are involved in complement. That's all you need to know about that. Here, this is just showing you class 1 versus class 2 molecules on the outsides of cells. This is where it would be like associating with this top part of it, associating with the actual antigen. Here, this is where we're presenting the antigen on the outside. So they look a little different, but they have similar functions. Class 1 is going to be, again, presenting antigens from inside the cell, class 2 from outside the cell. All right, telling cells apart. If we were looking at these cells and wanting to know the difference between these cells, one way we can differentiate is based on the cluster of differentiation, or CD. There's so many of them, there's a ton of them, but these are markers that are important in immunity, as well as the secondary signal. So if I come and interact with your MHC that's showing me some flu virus, and I'm a T cell, and I come in there and there's a CD marker that is also triggering at the same time during that binding process, that interaction, that's just reinforcing that this interaction is supposed to be happening and is happening correctly. So they work that way as well. Lymphocyte markers or their receptors. So B cells, we've already talked about how they can differentiate and make antibody. They're the only cells that will ever do that, by the way. There's no other cell in the entire existence of our bodies that can make an antibody. I don't want you guys to get too confused about that. So only B cells will be able to do that. T cells are going to be our cell-mediated immunity cells. Our antibody-producing B cells have a receptor on their surface called the B cell receptor that is basically an anchored antibody. It's basically what it is. It's just a membrane-bound antibody. It works the same as an antibody. And then the T cells have a similar type of molecule, but it will never be outside of the cell. It's only ever going to be anchored inside of the cell for those guys. They don't secrete them like the B cells do their antibodies. Okay, tissue macrophages are macrophages hanging out in the tissues. They're not circulating in the blood anymore, right? Monocytes are circulating in the blood. They get called into a tissue because of an infection, and now they become macrophages. So they're going to ingest pathogens. They're going to have an inflammatory response, dendritic cells. They're going to ingest whatever the antigen is in that area and go to the nearest lymphoid organ, and so it can be processed and presented to T cells, which again, T helper cells are typically our first step in a lot of this. And when we're activating this side of the immune response, the phase, that third kind of phase. And so antigen presenting cells include dendritic cells, macrophages, and B cells themselves. It can also include the monocytes, those are just precursors to the macrophages and the dendritic cells though, right? So those are proper antigen presenting cells. So B cells and T cells will proliferate and differentiate when challenged by an antigen. Whenever we're talking about cloning these cells, we're talking about proliferating that one lymphocyte. So if it is a lymphocyte that saw this specific exact shape on the outside of the flu, then it will proliferate and make a whole bunch. That is clone. Okay, so they're just being cloned, literally making the same one. All right, they're genetically identical, exactly genetically identical, and some of them will become memory cells. The T cells, and there are 3 types. Boy, okay, so T cells have 3 major types. The helper cells, which we've already begun to talk about a little bit. The regulatory cells, again, come on, no, if you can't guess what they do, they're basically going to help dampen the immune response, right? Keep it from going out of control. Cytotoxic T cells are going to lead to the destruction of infected host cells. You have a virus in your cells, multiplying in your cells, using your cell machinery. And now cytotoxic T cells are going to react to the fact that your cells are presenting on your MHC1, the virus molecules. And so that's going to allow the cytotoxic T cells to target them and kill those ones. So we can get rid of the infected cells. We can also use this to get rid of foreign cells. That includes, by the way, any tissue that you have had transplanted into you that is not an exact match for your body. So that's how that will die. We have the gamma delta T cells that are activated mostly by PAMPs and less specific response. But they can also be reacting to specific antigens. So it kind of does a little bit of both. I don't really need you to know much about them outside of that. All right, B cells will divide after being activated, turn into plasma cells and release antibodies into the blood. That will mark that antigen associated pathogen or whatever it is for destruction or neutralization, doing its little antibody thing. The innate response that will activate B cells. We have alert many components of the immune system to get active because a threat is on the way in a non-specific way. So they can start the activation process of the B cell, even though we don't know specifically what's happening yet. They can get that process started early. Antigen-specific T cell receptor will be on the outside of T cells. We'll also have those CD markers. CD3 will be assisting in the binding, the interaction with the MHC. CD4 and CD8, they'll also be involved in binding with MHC molecules, but CD4 is specific to class 2 and CD8 is specific to MHC class 1. So CD4 is associated with T helper cells. So we'll get to, don't worry. And then CD8 is associated with cytotoxic T cells because, like we said, MHC class 2 is going to be found on the antigen presenting cells. They were going around as sentries. They picked up some bacteria that came from outside of the cell. It came from outside of the cell. So that's important because we picked up our bacteria. We put stuff on our MHC class 2 markers that signals to the activating cells that we're going to be activating anyways, the T cells, that this stuff came from outside of the cell. I am an antigen presenting cell. I'm presenting this on MHC class 2 because this came from outside of my cell. So when it interacts with T cells, the T helper cells, which are always going to be our CD4 reactive cells, So our CD4 helper cells are going to be CD4 T cells, CD4 positive T cells, and we call them TH2 because they're interacting with class 2 MHC. That's going to go down the road of favoring, what do you think? Antibody, okay? The road that's going to favor cytotoxic T cells or destroying infected cells. You have any cell in your lung, got the flu in it, it presented that antigen on MHC class 1. It's going to trigger the activation of the T helper cell, which is a CD4 positive T cell. It is a TH1 cell because it's going to be interacting this time with MHC class 1. So our CD4 TH1 T cell that's been activated by presentation of an internal antigen presented on MHC class 1 has now been activated, and it can go and associate with that CD8 positive T cell that's going to be activated and to become active as a cytotoxic T cell and go destroy any of those cells that are infected with whatever virus. Okay, so that's another rundown of it. You see how we keep needing rundowns? Makes you worry when's the rundown going to stop? So the T cell circulation, they'll go between the lymph and the circulatory system, and they're just kind of always cycling through everything so they can get exposure to pretty much everything. B cells will mature in the bone marrow, where we know the T cells mature in the thymus, and then the B cells will circulate through the blood and kind of home to the lymph node, the lymph tissues in general, where they will take up residence, and they have immunoglobulins, which are their surface receptors that are basically antibodies on their surface of the cell. And don't tell them, all right, binding, immunologic, building. I don't know why I read that wrong. Diversity. Okay. Essentially, we have lymphocytes. Each naive lymphocyte bears an antigen receptor that recognizes a unique antigen. And how did it get this way? So we can potentially react to over 8 billion different antigens. I'm not joking. That's a real thing. So we can react to 8 billion different antigens. I'm telling you that there's these genes that are involved in every single unique protein that you have in your body. You have 20,000 genes, not 8 billion, sweetheart. You have 20,000. So how does this work? How can we get so much variability in these proteins? Because that's what antibodies are, they're proteins. How do we get so much variability in them that we could potentially react to literally any antigen in the entire universe? How do we do that? So what we do is we actually take our genes whenever we are creating our individual cells that could potentially react to these antigens. Whenever that is developing in the bone marrow, we will reassort those genes. Certain segments of them will come together, and there's a certain number of combinations you can achieve just by doing that. But our cells can also just put random nucleotides in there just to mix it up a little bit. So that's how we get that. Isn't that pretty crazy? Yeah, our immune system's that smart and cool that it's able to do that. So anyways, the stem cells that are in the bone marrow will essentially turn into whatever cells that they need to be, the white cells, the granulocytes, the monocytes, or the lymphocytes. The lymphocytes can be either B or T cells when they are developing, whereas the B cells will stay in the bone marrow. The T cells will migrate to the thymus to mature. Then they have their own little antigen receptor that we've made according to what I just told you about the genetics and the mix and matching, super cool stuff. And then when it gets to the tissues, it'll be equipped to only respond to that one antigen and nothing else. And so also in that process, we will be doing that clonal selection to help remove any self-reactive ones that we may have accidentally made when we're just doing that reassortment step. Okay, so this is just a picture depicting all of that. Yes, so this is how we'll get that kind of picture of reassortment. So we'll get sections of that DNA that will recombine, and we'll add in some extra nucleotides as well, just to give it some variability. And this is the part that's at the top. This is the part that's at the top. This is great. This is an immunoglobulin. Okay, whether it is soluble, floating around like as an antibody, or is attached to a surface, this basic shape here, this is an immunoglobulin. It has a top part, which is variable, and that bottom part, which is like the stem, like you might hold it by, and the colorful parts are the flowers at the top. That is the constant region. So they have purposes for why they're like that. So we essentially have 2 heavy chains and 2 light chains. You can see that these are what the heavy chains would look like in that setup down below. And then this is the light chains. They're just those smaller ones. Smaller ones are the light ones. Go figure with that. So one light chain gets bonded to one heavy chain, and then they get bonded to each other with disulfide bonds. So again, here is where sulfur becomes very important. This will create a symmetrical Y shape. All right, in that B cell receptor, our immunoglobulin that we're talking about here, we have antigen binding sites. They're pockets at the very end of the Y that will be formed by the light and the heavy chains at those variable regions. They can be so variable that they can fit, the whole universe's worth of antigens, potentially. So variable regions will be in those antigen binding sites because that's where we need the most variety, where we're actually going to be interacting with all these different bad guys. You want that going on in the end, like you want that to be the variable part, right? So constant regions, again, the amino acids aren't going to, they're what they sound like. I mean, they're pretty much always the same. They're not variable. These are not going to be, the part you hold it by, the stem, that part is not going to be undergoing a lot of changes. So they're going to have their set structure at the ends where the flowers are. Just imagine there can be all these different flowers that could potentially happen. That's the variable region. All right. T cell receptors are pretty similar. They have variable and constant regions as well as an antigen binding site with polypeptide chains that are going to make up the structure of it. T cell receptor is smaller and it's never going to be secreted. It's never going to become like the antibody. Antibody is only made by B cells, right? So this is that clonal selection theory. We're going to be removing, when we're, growing up in the bone marrow, we're going to be removing any cells that have created that variable region that we got mixed and matched with our DNA while we're making our cells. Any of them that develops to interact with self molecules that will kill those guys off so that they don't ever come up again. And then we've got this whole repertoire of these other cells that are non-self, hopefully, right? We will have those guys just lying in wait, always, one of each, right? One of each. So, basically, the right antigen comes along, interacts with that one cell will then proliferate a whole bunch, and some of those cells will differentiate into effector cells, which are going to do the job, like making antibody as plasma cells, or differentiate into memory cells, or maybe they'll be regulatory cells. They're all over the place, the regulatory ones. So immune tolerance is referring to our tolerance to any self molecules that we might have, and that's supposed to be removed through that clonal deletion that we saw in that previous step. Autoimmune diseases are thought to be caused by loss of immune tolerance or survival of the forbidden clones. They didn't get removed. There's some sort of failure in the whole process, or in a process outside of this that could have led to the whole scenario. It just kind of depends on how you got your autoimmune disease, and that can differ from person to person, even with the same diseases. So that depends, but that's the idea of how that's going to work. So next we have the presentation of the antigens. All right, an antigen is anything that'll provoke that immune response. An immunogen is technically an antigen that has been responded to by the immune system. So our immune system has recognized it as bad guy, okay? To be perceived as an antigen or an immunogen, you have to be a certain amount of foreign, a certain shape, a certain size, and you have to be accessible. If we have a B cell out there that could potentially react to an antigen that is way down deep inside of a protein and it will never see that, then that's not useful, right? So it has to be accessible. The terms antigen, immunogen are often interchanged. Yeah, we already mentioned that. So don't worry about knowing the difference. I don't care. So antigens have to be perceived as foreign. This can be reacting to whole entire microbes or parts of those microbes. Like when we talk about reacting to, you know, LPS, well, it's a similar concept here, okay? These could be cells or substances that arise from other plants or animals or creatures. They can also be molecules of complex composition, like proteins or protein-containing compounds that are able just to elicit the response. So it can be any number of these. Microbes or their parts, substances from other organisms, and then just molecules that can elicit a immune response, essentially. So we have good antigens. Good antigens. I don't mean they're good for you. I mean you have a good response to them. So what makes an antigen our response to it good? Okay. So what makes that happen? Their chemical composition, their context. So whatever signals were going on at the time that we encounter it, their size, so that can help. In general, larger ones are better. Large size, but is not sufficient to provoke a response, though. We still have to have the other things that we've mentioned in the last slide that I have to think about. So an epitope is a portion, like I mentioned earlier, a portion of the antigen molecule recognized and responded to by the lymphocytes. So the antigen is the book, the epitope is the sentence in the book. Okay. This is the primary signal that that molecule is foreign and not self, and we need to get rid of it. Haptens, these are little, tiny, too small to be reacted to by those immunoglobulins. They're just too small. You just can't react with it, you know, by itself. So we can have carriers link with them and then sort of carry them around, and then we can react to it, create antibody that sees that as foreign when it's linked to the carrier. So that when we see it again with our memory cells, they don't need the carrier back then when that happens. They only need it for that first sort of thing. So this is that hapten carrier situation. Here's our little hapten. The mouse will not make any antibody here. And then we have hapten molecules being attached to their carrier molecule here to make them bigger. And now we can associate with the antibody and start secreting antibody as a result. And that's great. And so we'll be able to do that whenever we see that molecule again in the future. Tetanus, by the way. Okay, tetanus. The tetanus vaccine works like that. All right, alloantigens, these are proteins and other molecules from another person that we see as foreign and react to as such. These can usually be due to cell surface markers that we see as non-self, even though in general, a microbiologist, well, a biochemist would look at it and say, this is human, this is not a bad deal. They're going to look at those markers and say, this is not us specifically, so we need to attack it. So this is also important, not just for organ transplant and grafting, which, but it's also important for transfusions, for blood transfusions. This is also why you would want to make sure your blood type is matched properly. Right, super antigens are essentially bacterial toxins that are extremely potent stimuli for T cells. They're going to activate T cells 100 times greater than ordinary antigens, which is terrifying, I think. And that can lead to an overwhelming release of cytokines called a cytokine storm. And as the body is reacting to those, this is going to lead to shock, and this is going to lead to death of cells. This is going to lead to potentially death of people as a result of this. Very quick. This happens very quickly. Toxic shock syndrome is included within this. So toxic shock syndrome is associated with a super antigen causing it. So, all right, antigen processing and presentation by our antigen presenting cells, our big heavy lifters when it comes to starting everything off right with our immune responses. We have the macrophages, the B cells, and the dendritic cells, the monocytes. So antigen presenting cells will grab that antigen and ingest it. So when I say grab and ingest it, we're talking about phagocytosis, right? And they'll digest it into pieces and then pass it out of its cell, either on its MHC2 or MHC1 marker, depending on where it got the thing from. So we get our little microbe and we eat it up from our little environment. We break it down into its parts. Some of those parts can interact with our MHC2 molecule because this one came from outside. So then we will present that in the MHC2 molecule and can interact with the T cells and go and, get them excited, get the T helper cells excited. So essentially now I'm presenting those on our MHC2, and here are our T helper cells with their little T cell receptor, nice and cute here. There's a little zoomed in area, so you can see all the CD markers that are associated with cells. Remember, CD4 is always a T helper cell. So these are going to be, interacting with their MHC2 molecules. So this newly activated T helper cell, this is going to become a TH2 because it is reacting to that MHC2. Okay, so that's important as well because that can help us keep track. And there's going to be signaling between these two. It's talking about interleukin, but there's going to be signaling between the two cells to let each other know, yeah, I got this. This does look like a bad guy. I agree. How about you go back out and fight? Or here, I found this. What do you think? This looks like a bad guy to me. And whatever. Let me help wake you up. So that sort of stuff. So now we have activated our initial T cell, our immature T cell. That is just, waiting to get activated. Now we've interacted with our antigen presenting cell and we're excited. And what's the first thing we do? Proliferate. So the first thing we do is proliferate. That's it. But then it will differentiate into memory cells. We already know what that does. It'll come back whenever we see the thing later. We have regulatory cells to dampen the immune response as needed. We have TH17 cells that are involved in inflammation. We have TH2 cells, right? And then we have TH1 cells, right? Depending on where they got their signal from, it will differentiate into TH1 or TH2. So TH1 and TH2. So both of these guys can stimulate macrophage activity to tell them to go back out. What does it do that? To tell them to go back out and go fight the good fight. So that's what's going on here. So we're stimulating our macrophages. So this whole process of having to get from antigen presenting cell, activate the CD4 positive T helper cell, differentiate into the T helper 2 cell, and then now it's going to interact with the stimulation of the macrophages to tell them to go fight the good fight. It takes time. It takes time. So this is why whenever they give you a PPD shot to check you for tuberculosis when you go work at a hospital, they'll tell you to come back in 48 to 72 hours. They don't tell you to come back in 20 minutes because it takes this long for this response to happen. So that's what we call a delayed type reaction. And then anytime you have a hypersensitivity that's delayed, like you see the thing and then maybe a couple days later, you start having the reaction after being exposed. That's what poison ivy does. All right, so our T helper cells can get activated by one of these. I'm trying to talk about B cells. I'm looking at them and saying T helper cells. These B cells can get activated by one of these T helper cells, or if they aren't reacting with them or have some sort of other signal that indicates they need to dampen the response, they can also help with the regulation. So it just depends. So we can activate our B cells with these guys. We can signal reproduction of the B cells and whatever it is. So you can see quite clearly that this TH response, the T helper response, it's incredibly important in all of the steps involved in driving the immune response. T helper is involved in step 1. It's like calling in the sergeant to boss the troops around. Okay, then we have here our CD8 cell activation. So we have our antigen presenting cell, and it will go and let's say it had a virus in it, like the flu, whatever. So it's presenting on its MHC1, right? So it's going to interact with the CD8 cells. And when we have activation from that antigen presenting cell, as well as from a T helper cell, which is also required, they have to have the signals from both of those sides. Now we can differentiate that CD8 cell, which is the precursor towards becoming our cytotoxic T cell. So we'll have a memory cell after that proliferation. We'll differentiate into memory cells and effector cells, ones that are going out to do their job. And that's going to be what's going on here on this side. So here's our T cell. It's going to make perforins and gram signs. What do you think that a perforin does? It perforates, it pokes holes, okay? So it's going to poke holes in the infected cells. And then granzymes are enzymes that are essentially going to be breaking down the contents of the cell once they get in through the perforin holes. And then those cells will undergo apoptosis because of all that breakdown that's going on. So that's how those work. So this is just an outline of all of our T cell subtypes. If you need to go back and review, which I'm certain you do, it's a great place to come to and look at their markers that are involved, as well as what they do for the body. All right, antibody production. First, we have our little binding of our antigen. Then we have our antigen processing and presentation on that MHC2 marker. So it came from outside of the cell here. That usually is what you would need the antibody for, things that are outside the cell. Right. So, and then the T helper cell, so Th2 typically now here, will be helping to activate that B cell, and they'll kind of talk back and forth with one another and recognize each other back and forth. And now we have that B cell activation. That B cell is now going to proliferate and then differentiate. Go figure. So proliferated and differentiated into 3 cells. We have our plasma, our memory, our regulatory. These plasma cells are our true effector in this case, okay? Yeah, right, with a mouse. Okay, so these are true effector cells, these plasma cells. They're going to produce antibody. The first antibody that's going to be produced in response to being stimulated by an antigen is going to be IgM, always. IgM is the first antibody that's produced, guys. IgM is the first antibody that is produced. It is a pentamer. That means there's 5, the Y looking guys, which I say was like the stem that you hold onto in the flowers, that Y out at the top with the variable regions of the antibody structure. It has 5 of those hooked together on the stem portion, the constant regions. So they're all, that's exactly what they look like in this picture. Okay, so they're out kind of like that. Why would it be like that? Because it allows the antibody to interact with a lot of antigens and pathogens all at once, and get that immune response going as quickly as possible, and to help boost reaction, from the other cells as well. So yeah, so we have those memory cells, and those will go into circulation, and so the next time that we see whatever this thing is, that we will be able to crank out antibody quickly instead of having to wait for all this whole process to happen. All right, cell mediated. All right, we already talked about these T lymphocytes. We have to have a direct involvement of the T lymphocytes. So that's why we call it cell mediated. It's complex. It requires MHC recognition before they can be activated. If that isn't there, because we can see this, the T cells that are going to recognize non-self MHC. So if it's being presented on something that is not our own MHC, that's why that has to happen. That's why that has to happen there, because this is the side of the response that would be responding to that, like projecting an organ is what I'm saying. So, sorry, I'm just making sure I'm not missing something. Cytokines, they make them. All these cells do. So cytokines, again, they are any chemicals these cells would be producing that are going to be involved in that immune response. So those are cytokines. T helper cells, they're going to be involved in regulating reactions to the antigens, including activating the B cells and the T cells, the cytotoxic T cells, right? Other T cells. And also activating macrophages, but also cytokines that we're making. Stimulation of the T helper CD4 cells by antigen and MHC complexes when we're actually presenting on MHC antigen will help differentiate them into what I have been telling you this whole time. So if it got it from inside, it's presented on MHC1, then it will differentiate into T helper 1, TH1. If it got it presented to it on MHC2, then it will differentiate into TH2, T helper 2. There's also the T helper 17 response as well. I mean, that's involved. It just depends on the cytokines and that the cells involved are secreting at the time. All right, our regulatory T cells, of course, these are also going to be CD4 cells. These guys are going to prevent any autoimmunity from happening, because when there's a lot of response going on to these foreign things, we can accidentally signal the body to attack self, unfortunately. So that is how a lot of autoimmune diseases are purported to possibly have come about. We also don't want to target, right? We don't want to target our normal biota, right? We don't want to attack those guys. We should see those as a, like we said, non-self, but okay, they're good guys still. Our B cells, they'll become activated to become plasma cells. They'll produce cytokines, they'll react to the cytokines produced by activating, T helper cells. And they will then make their own antibodies and their own cytokines as needed to dampen the T cell response if it is required. Just depends on how much signal and where and how. I keep wondering what's hitting my wall over here. And it's like, I have like one of those hammock chairs and it's storming outside. I keep thinking it's my neighbor. It's not. All right. Cytotoxic T cells. These are the ones that are going to kill other cells. These are the terrifying ones. So this is cytotoxicity, refers to the ability of these T cells to kill their target cells, which are going to be self cells. It doesn't have to be self cells, but the target cell. Cytotoxic T cells are going to be CD8 positive, and during activation, we have to recognize a foreign peptide that is complexed with MHC1, and then that will allow us to mount an attack on that target cell that's presenting that. All right, we talked about perforins. They are going to cause us to poke holes in the cells and allow stuff to leak out, which is bad enough, but it's also going to let us put the granzymes in. What are the granzymes in? These are enzymes that attack proteins of those target cells and then leading to apoptosis with exposure to these two. So that's how a cytotoxic T cell is going to kill their target. They're going to target virally infected cells, cancer cells, which are also bad from the inside, and cells from other animals and humans, which look to us like they're bad from the inside. All right, coming back to the structure of antibodies. Antibodies, this is our basic immunoglobulin molecule. So that's the basic foundation for an antibody, okay? We have those heavy chains and the light chains. We also have the FAB. That's just the top part. That's the Y portion. The part that is at the bottom, the constant region, that's the constant FC, okay? It's constant FC, C for constant. FAB at this area is F fragment is what it's supposed to be for, but fragment of antibody. This is the part that we care about with the antibody, right? Is the top part. I mean, let's be honest here. The FC is that constant region. So that's the structure of it. We've already talked about the heavy chain and the light chain and how they come together in their structures. So the FAB fragment is going to have those variable regions associated with them. It's also the special region of attachment between the FAB and the FAC that will allow for swiveling of the FAB fragments for better accessibility. The FC fragment, that constant region, is going to serve as an anchor to hold it into the membrane when it is acting as a B cell receptor. It'll be involved in binding to various cells and molecules of the immune system itself. Once we start secreting the antibody, that's what it'll get involved with. This is the function of antibody in general. This is what we've probably all been waiting for. What does it do? How does it help? Just keep telling us we need it. What does it do? So what it does is it can coat the surface of the bad guy. It can do something called opsonization, which means helping out with phagocytosis. Some of these guys, remember, they're going to have capsules or other things like that that can make it difficult. This is just going to make it that much easier for our macrophages to go back out, and they're triggered to go back out to eat them up, to destroy them. So that's opsonization. That's going to be anytime we're coating something with antibody or another molecule that makes it easier for the phagocytosis process to happen. This is also what happens with complement. Complement can lead to this as well. Then we have neutralization. Our antibodies are going to fill surface receptors on the virus or other active site on a microbial enzyme to prevent it from attaching normally. So very specific blocking action going on. We have agglutination, where our antibodies are going to interact between the pathogen structures themselves and cause them to stick together. And if they're stuck together, they can't do what they're trying to do. Here we have activation of the complement pathway. We already know this is the classical way to activate complement, right? learned about that already in the last chapter. So complement's going to come along and it's going to create its membrane attack complex to poke holes in cells to lyse the bacteria, break them open, essentially, so they'll leak their cell contents. So that's what the complement is going to be doing for that could be triggered by presence of antibody in the classical pathway. Now we have antitoxins, and antitoxins, these are going to be any type of special kind of antibody that neutralizes exotoxins. So now we're not talking necessarily about neutralizing the ability of a pathogen to associate itself with, surface marker. We're talking about something that they are secreting that could be bad for us, and then blocking that ability of that. That is what the tetanus toxoid vaccine. Tetanus vaccine is a toxoid vaccine, which is a bit of the actual toxin, and just the toxin, not the cell itself, that we are actually reacting to, because that's what gets us sick, is the toxin. So we get antibodies against that toxin, which is a hapten, by the way. We have to have a carrier there. But anyways, and we can block that now with our antibodies. So if you ever are exposed to tetanus toxin, that you will not get sick. Right, so this is just going to tell you guys that opsonization. This is going to be attachment of that antibody to foreign cells or viruses and expose epitopes to which they are bound to the phagocytes. So the phagocytes can come and eat them up more easily. Sort of marks them for eating. Certain antibodies have regions on the FC region for binding complement. It just depends on the antibody. So we already talked about that. We went through the whole thing. It's acting like I didn't show you the whole picture. And I definitely don't want to talk about basophils and mast cells. Okay. I mean, I should, but I'm not here. We'll wait till we get to the immune disorders. So here's our different types of our immunoglobulins that you can have: IgG, IgA. Which one is made first? IgM. I don't know why it's shifted this way, but it is, so sorry. IgD and IgE. And then where they can be found and their functions and everything. So IgM is this one here, the pentamer. That one is going to be the first one that is made in a response. Eventually, we want to gradually switch back over to making IgG. It's a faster response and a stronger response and a greater response. Whenever you are re-exposed to something like chickenpox, for example, then you are going to be producing IgG, not IgM, in response to reactivation of your immune system. Your immune system is so great. Your immune system is so cool that even if you do go over to a child that has chickenpox, well, I'm assuming you've been vaccinated for chickenpox or have had it already. I have had chickenpox. So if I were to go over to a child that actively has chickenpox and lick them, then I could go home and rest assured that my body will create so much IgG antibody against the chickenpox virus right away. I won't even know I ever licked that kid. Don't post that phrase on the internet, please. Okay. Anyways, moving on. Totally appropriate conversation we're having here. The other ones that work better based on their shape depends on where we're going in the body. So these guys here, the dimers, they can also work as monomers, but these are IgA, and they're very important with the mucous membranes. We'll learn about these as we learn about our diseases in this course as we work through it. But yeah, so those are the ones I really want to focus on: IgG, IgA, and IgM. IgD is just the B cell receptor. It's just the receptor on the B cells. And then IgE is involved in worms and allergies. That's, I guess, pretty important too, but we'll be talking about it on its own later. So titer is whenever we're trying to measure the concentration of antibody in your blood at the time. Basically, if you are at a certain point in the infection, how much antibody that you have made in response to that pathogen can be measured and tell us like how far along you are in the infection, or if you're still infected even. That can relate a lot back to that. And also how just in general, we can look at how certain people react to these antigens as well. That's the right thing. That's okay. All right, the primary response is going to be that first response. There is a latent period where we're just trying to, like I said, you're going to activate you're going to proliferate, you're going to differentiate. You have to do all that before you can do your job. So waiting on all of that to happen after being exposed to a pathogen can take time. So that's the latent period. Plasma cells will then produce the antibodies, serum titer will increase to a plateau, and then it'll taper off over weeks to months as the body realizes that there are no more invaders left. So it'll do that gradually, just in case there's any more hanging around. For the second response, which is the amnesiatic response, this will occur whenever you see the thing again, whatever thing it is, the chicken pox. This is that response that you'd be having. So it can be years later even. Then you can look at antibody synthesis and titer and everything there. You'll see it's way faster. You'll have IgG right away and a ton of it, and you can clear any potential invaders a lot faster, All right, primary and secondary response, just comparing those. You have response to your first antigen, and you're trying to just build up a response here. And then you're going to make your IgG and your IgM first, which is a weaker response, and then you're going to switch to IgG, and then you'll clear the infection finally. And then the second time that you see it, even years later, you're just going to start making IgG right away, and really not much IgM at all during that. Sorry about that. Let's move on. Okay, active immunity. In order to have active immunity, you have to have an individual receive an immune stimulus of some kind. So an antigen that could be from a natural source or an artificial source, right? So that means, a natural source would be like being exposed to a disease, and artificial would be like a vaccine. Passive immunity is whenever I'm going to give you like antibody or immune complex, and then you're able to survive. So you won't have any memory or anything like that. So we're giving you the result of somebody else's immune response. But that'll help you hopefully recover until you can maybe get your own, or even if you're trying to treat diseases, they use this. So immunoglobulins, that's what this is talking about here. That would be our, if we're using immunoglobulins, like you get bitten by rabies, and by rabies, you get rabies from a rabid dog, bites you, you're going to get your vaccine and you're going to get the immunoglobulin. Both of those have to happen relatively quickly in the exposure process in order to protect you effectively, because it's going to take some time for you to build up the immune response of your own. But in the meantime, we've given you that immunoglobulin to help you start fighting the infection right away. So that's what that's about. The immunoglobulin can act right away. You don't have to wait for that latent period. So that's the good thing with it. The other thing we can do is use immunoglobulins to infuse them into people and have them be targeted against cancer cells and similar sorts of things. We also have them target receptors to block, like let's say, imagine a receptor is a lock on your door. That's the receptor. And the key is coming along to bind with it, okay? Well, if you wanted to block that interaction, maybe just shove some putty in there, or weld it over, whatever it is. Immunoglobulins can do that basically by sitting on top of that lock so that the key can't get in. What I'm talking about here are receptors that would be activated if they could interact with TNF alpha, for example, a cytokine, which stimulates inflammation. If you have an inflammatory disorder like lupus or other autoimmune diseases, or other similar autoinflammatory conditions, you might want to block that inflammatory response. That's Humira. So now you learned how Humira works. So this is the idea of passive immunity. Those are all artificial passive immunity. A natural passive immunity might be like, how do you give somebody your antibodies naturally? Breastfeeding. So, so natural immunity, anything that would happen without medical intervention. If you start having medical intervention there, then we have artificial. So we've already just talked about that gamma globulin or immunoglobulin. Gamma globulin is the same thing. And then infusing that into people to, treat cancer and all sorts of, prevent inflammation and all sorts of things that can be useful like that. Okay. Of course, we can give these, like if you have Ebola, we treated some people that had Ebola that way. We took plasma from people who had survived Ebola, and and collected that, and then were able to concentrate the antibodies from that and provide it to people who were ill with Ebola and help save them. So that's something that happens too. And not just with Ebola, this happens with other things as well. That's a useful tool. So vaccination, that'll be exposing a person to something that is antigenic, but it won't be a pathogen. So that's the idea anyways, right? Sometimes, unfortunately, we do have some vaccines for viruses that are what we call, they are attenuated, which means they're not supposed to cause infection, but they can revert back to their wild type. And that is the case with polio vaccine. Unfortunately, that has been happening to people getting vaccinated for polio. So yeah, obviously it's important. One of the most important situations that we ever saw in vaccination in the history of all of medicine would be with Edward Jenner. And he noticed that milkmaids had nice, soft, beautiful skin, whereas other people were getting exposed to smallpox and they were having horrible scarring. So he looked into it and noticed that the cows had pox, and that was cowpox. So he scraped some of the cowpox off and inoculated his son with it, and then went and exposed his son to smallpox, and he was protected. So I don't know if you know this, but vacca is the Latin word for cow. Why do you think vaccine is named what it is? You learned a thing just now. Isn't that cool? Yeah, vaccines are named after that. So, that was vaccinia virus. That was cowpox that he used as a way to protect from smallpox, which is variola. So they're similar in structure, and we're able to protect whereas you're not risking, having this horrible scarring, disfiguring scarring from the cowpox. Yeah. So pretty cool stuff. Right. We know this is going to stimulate our primary immune response, prime our immune system to react against whatever the pathogen is that we could be seeing in the future. So that our secondary amnesiatic response will be triggered really well when we see it again the next time. Hopefully you don't see it again, but if you do, you'll be protected. So we have live attenuated cells or viruses. These are alive, technically, not killed. They are active, but they should stimulate strong immunity, but not be able to cause illness in the individuals who are being treated. And then we have whole killed cell or inactivated virus, which is what it sounds like, the dead thing of whatever you're vaccinating for to trigger the immune response. And then we have this one, they're saying unrelated viruses. So just rando virus that can carry the genetic material for components of microbes. So there was an adenovirus, I believe it was adenovirus, very common cold type virus. They gutted it and put the genetic information of COVID into it. And then that's one of the other ways that they were able to vaccinate against COVID whenever that whole spiel was happening. Or still, I guess. This is just going over a little bit more information, explaining how all these vaccines work. And I can see down here, this is the Johnson & Johnson vaccine. This is the one that is going to be involved with the harmless virus being gutted, and we put the genome of the COVID into it and then put that into your body, and your body responds to it because it sees the proteins that are going to be made by virus. It works similar to the mRNA, but we're using a virus to get it in there, basically, which is a more traditional way of dealing with things, but it is still pretty unique. So, right, so I don't know, here they're just talking about killed viruses versus live attenuated virus. And this is usually causing, the live attenuated just causing some mutations or something so that it can't actually cause disease. It shouldn't be able to cause disease in people. But yeah, so that can help with the immune response, of course. We see that with the MMR vaccine, the measles, mumps, and rubella virus. Flu vaccine tends to be killed vaccine if we're injecting it. If you get it in nose and in your nasal cavity, that's not the same. All right, vaccines that are based on microbial structures, essentially. So this is going to be the actual literal microbial surface structures. So they're spikes, they're toxins too, that counts, like toxoid vaccines like tetanus. All of those are going to be the microbial surface structures. It's not the whole microbe, right? And then we have DNA or mRNA in vesicles that will be coding for microbial surface components. So it's just like the virus one that we just saw, except in this case, there's not a virus. We're just putting it in a vesicle to keep it kind of safe in the meantime. But it should just be straight up, genetic material. Then we have surface subunits that are conjugated with proteins. What is conjugated vaccine? What does that mean? This will be usually we're going to conjugate the toxin or something like that onto another microbe's protein that we could be trying to react to. But anyways, it could make both of them more immunogenic, or make one that isn't very immunogenic more immunogenic because the other one is, and so on and so forth. So that just putting two things together and using the fact that we're responding strongly to at least one of them to help drive the reaction against the other one. So that's the idea with that. Immunogenic means like the strength, like your response against it. So we call those conjugated vaccines. Yep. So this is the idea of we have virus here. We have our surface antigen from the virus. We actually use that surface antigen itself to plug into your body, and then you have a reaction against that with your antibodies. This is how it would be Hep B works. If you are going to be, we have your actual mRNA. This is the mRNA vaccines that we were talking to you guys about. mRNA, just protected by some lipids just to keep it safe. Inject it into a person, their cells start making the viral protein and recognizing it as foreign. So now we have antibodies that are going to be attacking those foreign molecules the next time they see them very quickly. This is Pfizer and Moderna. Then we have a virus or bacteria. They have their little surface antigens, but they're not going to elicit a very good response, we've found out. So we'll put them with some other thing to make them create a stronger response. So this works with Haemophilus influenzae type B, or Hib. That's a conjugated vaccine that is associated with meningitis, as well as pneumonia, that kind of thing. So booster shots, of course, we need these to administer to keep up your response. We don't want the cells dying off and thinking that they're never going to see this thing again. So we get booster shots in order to keep your immune system on its toes. If for some reason there is a vaccine that can provide protection for a lifetime, then you wouldn't need a booster shot. But this is not going to be the case for a lot of the viruses and stuff that we would be looking at. Sometimes you just need a boost, just remind your body that you need to keep these things around just in case times, right? So the COVID vaccines, we already talked about the mRNA codes for a surface protein, the spike actually of the COVID virus. And the proper mRNA ones are going to be coated in just a lipid vesicle. So it's just a lipid vesicle, not a virus. It's not anything else, just a packet. It gets inside, we start producing the S protein, the spike protein in our cell. Our body sees that and starts making antibodies against it. And now we have protection. The other ones, the Moderna, no, the Johnson & Johnson was made with the viral one. So we just took a naked adenovirus and popped some COVID DNA in there, and then it worked the same way essentially after that as the other one. So other ways that we could look at trying to fight disease, maybe using nanoparticles of certain surface proteins of these organisms to try to get a response against those. Okay, yeah, so I figured it out. So basically the nanoparticles will be made of the surface proteins or whatever, or at least mimicking them. But anyways, you wouldn't need the cells of the human that you're injecting this into to do any work of translation or modification of this. It would just straight up work just as it's delivered. So that's the idea there. Then we have gene editing techniques like using CRISPR, which we've talked about previously, where we can engineer our own B cells. We talked about all that gene rearrangement, right? We can actually engineer B cells with CRISPR so we can express exact antibodies that we want to design to target this exact thing and have this kind of response. So that's pretty cool stuff too. How do we administer vaccines? Either subcutaneously or intramuscularly, which you need to understand those are not the same thing. Intradermal, which is, again, like you also have those little patches you can put in areas and with these little teeth on them that poke into the skin and deliver whatever the antigen is that way. Often can use those for allergies. We have oral or nasal vaccines that are not very common, not used commonly, usually used in special cases. An adjuvant is a term that refers to a special binding substance that will be added to some vaccines to enhance immunogenicity. So to increase the likelihood that you're going to respond strongly to whatever it is we're injecting into you. It will also make it so that the antigen and everything that is going into your arm when we're injecting you will last longer, so we can give a prolonged response to actually build up some great memory. And that can sometimes, depending on the adjuvant, facilitate contact with the antigen presenting cells and the lymphocytes that are going to be involved in creating that memory response. So common side effects of vaccines are going to be local reactions, of course, soreness at the injection site, for example. Flu-like symptoms are common. That is caused by the immune response. That's not caused by being sick. That's caused by your immune response. So if you're sick with the flu and you're feeling tired and worn out and you're just sore and you got this fever, that's your immune response doing that, by the way. So when you go to get a flu vaccine and you feel that way, you didn't get the flu. Your body's reacting to it. It's exactly what it should be doing. So just keep that in mind. Anyways, most common serious side effect, there was incidents of anaphylaxis in response to COVID vaccine. Not common, 5 out of a million. So vaccine misinformation, things that get spread around that are not correct. This is considered to be a huge problem as far as maintaining health globally. So misconception, this is what I was just saying. Oh my gosh. Okay, individuals can get the flu from the flu vaccine. No, it is dead. Guys, no, you cannot. So you got a fever though? You got sore from it? Yeah, like I said, like good. That's your body reacting. That's exactly what it should be doing, so that the next time when you see it, your body won't have to react that way. It will just automatically spit out IgG and you won't even know you're exposed. So it's okay. That's okay. There's also that misconception, of course, this was the most popular one. This is the misconception that there is mercury in the measles vaccine that can cause autism. Number one, measles vaccine never had mercury in it. And number two, it does not cause, the measles vaccine anyways, it does not cause autism. There have been a lot of studies done since that paper was published, and that paper has since been retracted because the doctor that published it admitted that it was false information. So there you go. You don't have to worry about that. That's lies. And I didn't write a question of the chapter. I didn't write a question of the chapter, guys. I'm so sorry. I apologize. So your question of the chapter, I guess, would be, I want you guys to list for me the steps that your body will go through from the moment it sees an antigen, like the flu virus or, I don't know, a bacteria of some kind, until it would be, cleared essentially. And so give me that explanation. That's what I want from this one. And that's it for this chapter. And I'll see you guys next time. Bye-bye.